Thank you. Good morning. My name is Sisir Dal. I am PhD student here at Otago University. I'm the last speaker, and I'm concluding this session with a more clinical type of presentation. And the topic of my presentation is characterization of intraplaque calcification in Mars imaging with histopathological correlation. And I'm presenting this presentation on the behalf of Mars Collaboration and Mars team. Atherosclerosis is the major cause of morbidity and mortality worldwide. 1 in 3 deaths occur due to atherosclerosis or related complication. It's the disease in which plaques build up in arterial wall, which eventually get calcified. And it was found that amount of calcification in the arterial wall determine the severity and outcome of this disease. For example, amount of calcification in the coronary artery can predict heart attack. Amount of calcification in the specific location of carotid artery can predict stroke. Thus, the quantification of arterial calcification has huge clinical significance. Currently, the imaging modality of choice for quantification of calcium, measurement of calcium is computed tomography. However, measurement of calcium with computed tomography is energy dependent, and depending upon the energy of X-ray beam, computed tomography can underestimate or overestimate the calcium. Thus, the objective of my study is to identify an appropriate energy of the X-ray beam for the precise quantification of vascular calcification using Mars spectral photon counting CT, which can generate images in multiple spectrum and compare them, validate them with the histopathological reference standard. Methodology. First, the calibration phantom or scan to find the optimal linear relationship between different concentration of hydroxyapatite and CT output signal in Hounsfield unit, forming the calibration curve. Appropriate imaging protocol were set up. Voltage as 118 kV, 4 different energy beams, 18, 30, 45, and 75 keV. 10 excised carotid artery with atherosclerotic plaque were randomly selected from the pool of stored specimen. They were scanned with a Mars scanner. Thereafter, histopathological slides were prepared. 2 independent consultant vascular radiologists were asked to measure 18 different calcific regions in 4 spectral images, 1 material decomposed images, blind to histopathology, blind to each other in 2 different settings. And the results were compared with histopathological reference standard. Two histopathological staining protocols were used. Amatoxylin eosin, calcium is measured in the histopathological slide, then compared with Mars spectral images and the Mars material decomposed images. Another protocol is the Alzarin rate protocol. This is calcium specific protocol. And again, the calcium area is measured in the histopathological image and compared with different Mars spectral and the material decomposed image. To ensure the reproducibility of the result, possible sources of biases were identified and tried to address. Selection bias was addressed by simple randomization of the specimen. As a PhD student, as a Mars employee, I might have a confirmation bias. So we recruited 2 independent radiologists for the image interpretation. Observational bias is reduced by the interpretation of Mars imaging blind to the histopathology. And finally, the role of chance is reduced by asking, interpreting, evaluating radiologists to evaluate it twice. So we had 4 sets of data to evaluate from. This whole study was done with a null hypothesis that there is no difference between Mars and histopathology while measuring intravascular calcification. P-value was set of less than 0.05 for the statistical significance with 95% confidence interval. First, the data were normalized with the log transformation. and hypothesis was tested with the student t-test. Limit of agreement between Mars and histopathology was found with the blind Alpine plot. And finally, the reliability of evaluating radiologists were done by intra-class correlation coefficient. And it was found that there was a significant difference between Mars measurement and histopathological measurements in the lower energy bins, especially below 45 keV. And as you can see in this upper two row, the p-value consistently less than 0.05. Material decomposed images and images more than 45 keV were most appropriate for the quantification of calcium, with p-value consistently showing more than 0.05 in these three rows. There is slightly better limit of agreement between Mars measurement and histopathological measurements in higher energy images, more than 45 keV, as compared to material decomposed images. And finally, intra-rater reliability or intra-observer reliability between two radiologists were excellent with the intra-class correlation coefficient of 0.9. Similarly, inter-observer reliability between radiologists was again excellent with the intra-class correlation of 0.9. Thus, in summary, amount of vascular calcification indicate the disease burden in specific arteries. Material decomposed images and images of higher energy bins are most appropriate for the quantification of vascular calcification. And at last, the evidence we produce in this study shows a translational ability, because we are now using this evidence in our human imaging. Thank you.